Hello, good evening. My name is Rakesh Jalali. I work here in Chennai and I am currently the president of the Immuno-Oncology Society of India. On behalf of the Executive Committee of Immuno-Oncology Society of India and from all of us, it gives me great pleasure to welcome you all to today's webinar. And it's an extremely interesting topic chosen, basic biology to cancer immunotherapies. Now, several of us a few years ago came together to launch the Immuno-Oncology Society of India with a lot of enthusiasm, zeal, and expectation. And I have to say that it's been a great pleasure to be involved with the IOSI and to see its excellent growth over a period of time. And every year, as many of you know, we hold an annual conference, typically in January, and very successful and well attended. This year, also in January, we had an annual conference in Bhubaneswar, and last year was at NCCS Pune, led by Dr. Girdhari Lal, this time by Dr. Ghanshyam Biswas, and was an outstanding success with ultimately, of course, Professor Jim Allison, famous Nobel laureate. That was the crowning glory that he could attend along with Dr. Padmani Sharma and others, which exemplifies the niche that the IOSI has made globally, such that they felt the need to come down in person to speak with us, to interact with us, to the scientific community as well as to the medical community. In the same vein, the executive committee decided in Bhubaneswar to launch a webinar series because all of us know how important it is to understand the science behind this rapidly changing field, how the impact has taken place into the clinical practice, but also one must also understand that there is a bit of over expectation and hype also. So, and the field is there. The more you know, the more you realize how many more hurdles have to be overcome. And this will come only with the collaboration between the basic scientists, the bench, and translating into the bedside. So it was decided that there will be the first webinar will be exclusively on the biological aspects, which is the fundamental pillar of the whole story of immunology, but also in particular with the cancer immunotherapy. And I must compliment the executive committee and in particular Dr. Girdhari Lal, who was very kind and very gracious to take up the challenge and to do the first seminar. So hopefully this will be the first of many seminars and webinars to come. And the next year's IOSI meeting is in Chennai. Already we have started a little bit work on it in January. In a couple of weeks or 2 to 3 weeks, you will get the information. But primarily towards today is on the webinar. And finally, I would like to also tell that the forum encompasses all the basic scientists, translational researchers, whether they are directly or indirectly related to the field, because we have to come together to push the field, and the India also has to contribute to the global scientific platform. And it's about 80 to 100 basic scientists are at the moment members of the IOSI. The membership of IOSI is very simple these days, and please do go to the website, and we're delighted to have you as well as your students, as well as your researchers and trainees to become the members such that they can benefit from the collaboration and provides the platform. We also have some prizes for young people, young faculty to be able to travel to abroad, and we would support them financially if their abstract is accepted in a major immuno-related topic in any of the major conferences, whether it is basic science or clinical oncology. So with this background, may I just request my colleague, my dear colleague, Dr. Girdhari Lal, who is currently the Joint Secretary, IOSI, Program Director Scientist at the National Center for Cell Science, Pune, an amazing conference he had last year, and he is the program director of today's webinar as well. Dr. Girdharilal, kindly please take over to give us the background of the webinar, the topic, as well as introduce the speakers. Thank you.
Thank you, Dr. Jalali, for giving a big start of this meeting, webinar series, telling about what is the importance of this particular society, what we do. And on the same line, we actually decided to have something on basic immunology to learn biology of the immune cells and how these immune cells behave in the tumor microenvironment or in circulation. So we have on this series, that is the first series we starting today, the Dr. Silpak Chatterjee is going to talk to us how immune cells actually behave in the tumor microenvironment, how their metabolic activities are altered, and he has done wonderful work on this line. So thank you, Dr. Chatterjee, for accepting our invitation. And then we have another speaker, second speaker, Dr. Sudha Kumari. She is going to talk about how immune cell decide to touch and what is the molecular touch they actually require to work on. They're not just going to the microenvironment, they decide to touch not just tumor cell, other cells, and how this touch or untouch and the context of touch dictates their function and also in terms of anti-tumor immunity. So let's get started. Let me have the pleasure to invite Dr. Silpak Chatterjee. Silpak, can you hear us?
Yes, I can hear you. Yeah.
Right. So Silpak is a scientist at in Kolkata. It's called the Indian Institute of Chemical Biology, and he has done wonderful work. And he has done work from his PhD days, and he did his PhD from Jadavpur University, especially working on tumor-associated macrophages, and we know another cell, MDSCs, and then move on to the Medical University of South Carolina, and then he started working on adaptive cellular therapy. And now currently his lab at IACB is looking at the metabolic alteration of immune cell in the tumor microenvironment, especially focusing on T cell, what guide them to take advantage of their own metabolic profiling, and what are different ways we can alter them so that they can efficiently work in the tumor microenvironment. And today he's going to talk about, he's actually got a lot of good paper, and his recent paper actually is very interesting paper. I will urge also to the audience to read about it, and I'm sure he's going to discuss his work today on that line, how one of the molecules, CD38, and its metabolic profiling. And he is going to discuss today on immunotherapeutics modulation of T cell exhaustion in solid tumor. And we know what T cell exhaustion and what is the trouble we see in terms of immunotherapy, especially in the tumor microenvironment. So with this brief introduction, I will invite Dr. Chatterjee to load his slide, share his slide, and then start the presentation.
Thank you, Girdhari, and thank you, the IOC team, for the invitation. Just bear with me. Let me share my slide. So
you may have to click on share button.
Yes.
In share button, and share the whole screen so that it should be visible.
Okay, somehow. Just bear with me for a couple of seconds, because somehow this one it's not working. showing. Okay.
Very great.
So I'll go to the desktop meeting.
Right, double click.
Yeah. So can you see my slide now?
It might delay. Right now we can see your screen. Go to presentation mode and see that if that works. Right now we don't see your slide. You see your screen only. If you are on PowerPoint, go to full screen.
Yeah, I have. Yeah. So okay, and
Oh yes, now we got it. Perfect.
okay. I'm extremely sorry for the delay. Actually,
No problem. These are technical glitches sometimes.
yeah, because the other uh, laptop was not showing. But anyway, thank you, Girdhari, and thank you, Jyoti, for the invitation. And as the title said, so I'll be mostly talking about the T cell exhaustion. And some recent observation from our lab, how this T cell exhaustion can be modulated to improve their anti-tumor response. So as you know that T cells are the major hindrance for the successful immunotherapy of cancer in the immune checkpoint blockade therapy. And yeah. So as if you see the cancer immunity cycle, so when there is a tumor growth, obviously there will be infiltration of the innate immune cells, and these innate immune cells actually carry the antigen to the draining lymph node and license the T cells. And these T cells, after licensing, they go back to the tumor site and try to kill the tumor cells. So now if you see this, how this licensing happens, you can see that obviously when the T cells see the antigen in the context of MHC, the cognate antigen, and when there is a co-stimulation happens, then these T cells get activated. And when they get activated, there is a clonal proliferation of these T cells, and these T cells start producing different effector molecules like interferon gamma, perforin, and ganzyme. And these are the effector molecules which can directly kill the tumor cells. So what happens in the normal scenario when there is a acute infection, these naive T cells, when they see the antigen, they become effector in nature. And after resolution of the infection, most of the T cells die by apoptosis, but some of the T cells persist as a memory T cells. But if the antigen persists, the antigen load persists, if the infection persists, then instead of taking this route, the T cells go to the another route, and these T cells progressively acquire different immune checkpoint molecules and become exhausted. So as you know, these immune checkpoint molecules, mostly this PD-1, LAG3, TIM3, these are the immune checkpoint molecules. And this T cell exhaustion is a progressive phenomenon where the T cells progressively acquire these immune checkpoint molecules, and then they become dysfunctional in nature. They stop producing these effector cytokines, the interferon gamma, TNF, IL-2. They become non-proliferative in nature and become highly susceptible to apoptosis. So now to rejuvenate these exhausted T cells, which are expressing these immune checkpoint molecules, the immune checkpoint blockade therapy has been introduced in the clinic. And very simply, this immune checkpoint blockade therapy, how it works. So if you see that in the periphery, what happens if the T cells are functional? These T cells will produce these cytokines once they see the tumor antigen, and the tumor clearance will happen. But eventually, in the tumor microenvironment, the T cells become dysfunctional, and this dysfunctionality happens basically due to the expression of this immune checkpoint, as I said, like PD-1, CTLA4, and these molecules ultimately dampens the effector function of these T cells, and they stop producing these effector cytokines. As a result, tumor progression happens. But now, if you add the immune checkpoint blockade therapy to block this interaction between this PD-1, PD-L1, or block this using the anti-CTLA4, then what happens? You can actually rejuvenate these T cells again, And these cells again start producing these effector cytokines, and eventually the tumor regression happens. So, as you know, this immune checkpoint blockade therapy has yielded remarkable clinical success in the clinics. And if you see, for FDA has approved a lot of almost for every cancer there is immune checkpoint blockade therapy. But unfortunately, still now this therapy is responsive to a very small percentage of patients. Only the responder is only 20 to 30%, but the majority of the patients, which are who are like 70 to 80% of the patient, they do not respond to this immune checkpoint blockade therapy. So this actually incited various research questions. What are the T cells composition in the tumor microenvironment? Why these some of the patient do respond to this therapy? Why some of the patient fail to respond to this therapy? And so basically there is a interest to understand what is the T cell composition which actually respond to this therapy. What is the T cell composition in the tumor microenvironment? And recently, it has been shown that these exhausted T cells in the tumor microenvironment is actually exist in a different exhaustion states. And these exhaustion states actually determines whether these cells will respond to the immune checkpoint blockade therapy or not. And very interestingly, it has been shown that broadly you can categorize these exhausted T cells in 4 different groups. So like a progenitor 1 or progenitor 2 of exhausted T cells, they are usually responsive to the immune checkpoint blockade therapy. But as these T cells progressively enter into the intermediate exhausted state or terminally exhausted state, they completely lose their ability to respond to the immune checkpoint blockade therapy. And several groups have reported that one of the important transcription factors, which is called TCF1, this TCF1 actually regulates the self-renewal capacity of the T cells and actually determines whether the cells will respond to the therapy or not. So in the progenitor exhausted cells, which actually respond to the immune checkpoint blockade therapy, they have a very high expression of the TCF1, and the TCF1 is encoded by the genes called TCF7. Okay, so as these T cells enter into the terminally exhausted state, they lose the TCF1 expression and eventually become non-responsive to the anti-PD-1 therapy. So in our lab, we started to answer this question or ask this question: what is the composition? What is the signature of these terminally exhausted T cells? Okay. So as you know that in Indian context, to get a lot of patients who are actually getting this immune checkpoint blockade therapy, it's very difficult. So we started with a very cheaper version of this immune checkpoint blockade therapy. That is a very age-old immunotherapy called the BCG immunotherapy, and which is given to the non-muscle invasive bladder cancer. So what we did, we took the non-muscle invasive bladder cancer, took the tumor tissue and the blood before the surgery, and after that they were in the BCG immunotherapy, and we followed up this patient for 2 years and see what is the recurrence of this patient. And when we collected the tumor tissue from this patient before this BCG therapy, we actually tried to understand what is the immune contexture in the tumor microenvironment, or the T cell contexture in the tumor microenvironment. So what we did, we took the T cells from the tumor side, and using the flow cytometry, we get it using the 2 different immune checkpoints, very important immune checkpoints. One is CD38, another one is PD1, and then tried to understand whether this population are expressing only one immune checkpoints or they are expressing multiple immune checkpoints. And if so, which population is actually correlating with the responsiveness of to the BCG immunotherapy. Okay. So what we found that in the tumor microenvironment of the BCG or the NMIBC, there we could find 7 different clusters of the T cells. So some of this cluster like express only PD1, some of these T cells cluster express only CD38, some of this cluster express both PD1 and CD38, some are expressing PD1, CD38 and TIM3. And once we have this data, we then have this follow-up data from this patient and try to correlate which of this population is actually correlating with the responsive to the therapy. And very interestingly, what we found that the patient who did not recur, or the non-recurrence group versus in the recurrence group, there is a huge upregulation or a huge enrichment of this population, which is expressing both or which is expressing all the three immune checkpoints like PD-1, CD38, and TIM3. So this is the population which actually were threefold higher in the patients who actually did not respond to the BCG immunotherapy. So then we actually tried to correlate this population with the very well-known EORTC recurrence score. So EORTC is a European Organization for Research and Treatment of Cancer, and this score actually predicts the risk of recurrence in the patients with NMIBC. Okay, so what we found that among the all population which we assessed in the tumor microenvironment, the EORTC recurrence score was very positively correlated with the population which actually expressing all the three immune checkpoints. So this suggests that the patient who had this population from the beginning, this PD-1 positive, CD30 positive, and TIM3 positive CD8 T cells in the tumor microenvironment, they are kind of a poor responder to the BCG immunotherapy. So when we were working on this project and we tried to understand what are the literature suggest in the other patients and these where these studies try to understand what are the immune signature in the tumor microenvironment, we actually came across different studies which are already published in the literature. And what was common between all the studies and in our study was that all these studies, they have reported a particularly interesting molecule called CD38, and they have shown whenever the CD8 positive T cells in the tumor microenvironment had a higher expression of this molecule called CD38, somehow they are not responding in a good to this immune checkpoint blockade therapy or other immunotherapy. So then we tried to understand or ask the question whether this CD30 is somehow involved in making the T cells terminally exhausted, and this molecule whether related to the non-responsive to the immune checkpoint blockade therapy. So first, to understand this question, what we did, we took the real patient data or the patient who actually went through this immune checkpoint blockade therapy. So this is a publicly available data set, the single cell data set. So here, total 32 metastatic melanoma patients were taken, and they were actually treated with the anti-PD-1 and anti-CD, or either anti-PD-1 or combination of anti-PD-1 and anti-CTLA-4. So before the treatment, the tumor tissues were collected and single cell RNA sequencing was done. And after the treatment, they have segregated this group between responder and non-responder. And in each group, they also took the tumor tissue and did the single cell RNA sequencing. So what they found that yes, they found 5 different clusters of T cells present in the tumor microenvironment of this patient who are going through this immune checkpoint blockade therapy. So now, since it is a single cell RNA sequencing data, you can actually go into each of this cluster and see what is the transcriptomic signature of this cluster. And since our the interest was to understand which of this molecule or which of these checkpoint molecules is actually correlating with the immune checkpoint blockade therapy responsiveness, what we did, we tried to understand this thing in this patient cohort. So what we found very interestingly, if you can see this particular data where this is against the CD38 positive, CD8 positive T cells. So this is the responder group pre-immune checkpoint blockade therapy, and this is the non-responder group pre-therapy. And these two are a responder and non-responder post-therapy. So if you can see, the CD30 positive, CD8 positive T cells are highly abundant in those patients who actually did not respond to the ICB therapy. But this was not true for the other reported immune checkpoints like PD-1, although the PD-1 was high in the early patient, but there was no difference between the post-therapy. And same for the TIM3, LAC3, TIGIT, and others. So what we found that only CD38 is nicely correlating with the failure to the immune checkpoint blockade therapy. And then we tried to understand that whether CD38 is actually correlating with the molecules which are people have reported to be associated with the good responsiveness of the T cells. And if you can see that CD30 is actually negatively correlating with all the molecules that people have reported that has been shown to be negatively or positively associated with the checkpoint or effector function of the T cells. So if the CD38 is high, it is actually negatively correlating with all these molecules. And one of the top molecules was TCF7, which encodes the protein called TCF1. And people have reported that if the TCF1 is not there, then the T cells lose their responsiveness to the immune checkpoint blockade therapy. So since I'm a basic biologist, so my interest was to understand that mechanism behind this purpose. So why CD38 and how CD38 is regulating this whole thing and non-responsiveness to the therapy. So next thing what we did, we actually took the human T cells from the peripheral blood, normal human or from the healthy volunteers. And then we activated the T cells for 3 days and then subjected them to the chronic stimulation or the acute stimulation. So what is the difference between these two things? That people have reported that T cell exhaustion results from the chronic stimulation of the T cells. So if the T cells are continuously put into the stimulation, then the T cells eventually become exhausted. And this is same thing happens in the tumor microenvironment. So to mimic these things, we actually did the same thing. We took the T cells, activated, and then either chronically stimulated them for 8 days, or we activated them and put them in the cytokines and did not touch those cells for another 8 days. And then we took these T cells out and did the single cell RNA sequencing to understand how CD38 is regulating all these phenotypes. So if you can see that the single cell data, that this acute, the transcriptomic profiles of the cells which are acutely stimulated versus which are chronically stimulated is completely different, suggesting that chronic stimulation led to a different transcriptomic landscape altogether into the this exhausted T cells. And then what we did, we took this chronically exhausted cluster and reclustered them. And after reclustering, we found 5 different clusters, starting from cluster 0 to 4, and then tried to understand which cluster is expressing the CD38 and which cluster is expressing PD-1, and what is the association of this cluster with the TCF1, which is well-known regulator of the responsiveness to the therapy. And if you can see that this cluster 0 and cluster 4, they have a very high expression of the CD38. But very interestingly, this cluster do not express TCF7. But on the other hand, the cluster, these 3 clusters, they have a high expression of the TCF7, but they don't express CD38. So this suggests that somehow this expression of CD38 is negatively correlates with the TCF7 expression. So then what we did, we actually tried to understand the transcriptomics, whole transcriptomics profile, and did the histogram heat map analysis. And if you can see that this cluster 0 and cluster 4, they express all the molecules which have been reported to be associated with the exhaustion, and these molecules are associated to the or negatively correlates with the immune checkpoint blockade. So then going forward, we tried to understand really this CD38 do not respond to this therapy, or there are some other molecules which are associated with this non-responsiveness. So for that, what we did, we actually took the female mice. The female mice is a TCR transgenic mice where all the T cells express the receptor which is against the tumor antigen. So we took the T cells out of from the spleen of these TCR transgenic mice, and these T cells were chronically exhausted. And then after 8 days of chronic exhaustion, we sorted them based on the CD38. So we took either the CD38 high population or the CD38 low population, and then adaptively transferred into the mice which is bearing the B16F10 mouse melanoma. And this is to note that this B16F10 mouse melanoma is a PD-1 resistant tumor model. And once we did the adaptive T cell therapy of these cells into those mice, we either treated those mice with the anti-PD-1 or with the isotype control, and then try to see which population is actually responding to the checkpoint molecules. So if you can see here that where this CD38 high population was injected, these T cells, they failed to control the tumor. And when we combine them with the immune checkpoint or anti-PD-1 antibody, again, these cells did not respond to the anti-PD-1 antibody. On the other hand, when we injected or adaptively transferred CD38 low population, this population actually controlled the tumor very well. And also, when the anti-PD-1 therapy was combined with these T cells, the PD-1 therapy, the cells responded to the PD-1 therapy, suggesting that CD38 low population is somehow able to respond to the anti-PD-1 therapy, but CD38 high population somehow lose the ability to respond to the immune checkpoint blockade therapy or anti-PD-1 therapy for this purpose. So then we tried to understand the mechanism, how CD38 is actually regulating this thing. So for that, what we did, we took the two approach. We either knock down CD38 in the T cells using the shRNA or using the pharmacological inhibitor, and tried to see which factors is actually getting mostly affected by knockdown or by inhibition of the CD38. So we checked the PD-1 and TIM3 expression, although TIM3 expression was significantly down after the CD38 inhibition, but we did not find any changes in the PD-1 expression after CD38 inhibition. Then we checked the effector cytokine production, which actually determines whether the T cells are functional or not. And if you can see that CD38 inhibition actually improved the effector function of the T cells, as the interferon gamma production and the TNF alpha production in both knockdown cases and the inhibitor treatment cases was substantially improved. But most interestingly, what we found that inhibition of CD38 can restore the TCF1 expression in the T cells. And this restoration, we thought that this restoration is actually important to give them a stemness kind of phenotype and to improve their self-renewal capacity. So to test this further, that CD38 is indeed responsible for all these things, what we did, we again we took a mice model, and what we did, we took either the normal female mice or the CD38 knockout female mice. So in the CD38 knockout female mice, all the T cells, they do not have any CD38 expression. And as I said, these female T cells are TCR transgenic T cells, and this TCR is against the melanoma antigen GP100. So we took the spleen from both these mice, activated them, and then adaptively transferred into the mice bearing the P16 melanoma, and then give the anti-PD-1 therapy. And as we expected, we found that yes, when we took CD38 knockout female T cells, these female T cells did really better as compared to the wild type female T cells in controlling the tumor. And knockdown of or knockout of CD38 actually make this female T cell responsive to the anti-PD-1 therapy. So this suggests further confirm the fact that CD38 is actually important to determine whether these T cells will respond to the anti-PD-1 therapy or not. So as I mentioned, that we thought this the whole mechanism is actually regulated by the TCF1, because when we inhibited CD38, the TCF1 level was restored in the T cells. So we thought that is it the TCF1 which is regulating the response when the CD38 is gone? So for that, what we did, we again took the two mice, female and female CD38 knockout mice, and took the spleen out of it. isolated the CD8 T cells from there, and in one CD38 knockout group, we actually did the TCF7 knockdown. So in those T cells, CD38 is not there, but at the same time, we knock down TCF7, that means TCF1 is not there. And when we adaptively transferred these T cells into the mice and did the anti-PD-1 therapy, we found that yes, if we have CD38, they could control the tumor, but as soon as we knock down TCF7 from the CD38 knockout T cells, they lose the anti-tumor potential and they lose the ability to respond to the anti-PD-1 therapy. So it suggests that CD38 axis is actually regulating through the TCF7, and that by that way it actually regulates or determines whether these T cells will respond or not respond to the anti-PD-1 therapy. So to further understand the mechanism, what we found that CD38 is actually NADase. It actually break down NAD and from the cyclic ADPR. So it has been reported that whenever CD38 is high, the NAD level is low, and that's why I have not shown all the data. What we have found that the metabolic fitness is another important factor which actually regulates the responsiveness of the T cells to the therapy. And since CD38 regulates the NAD level, and if the CD38 is high, the NAD level will be very low in the T cells. And when the NAD level is low, this actually impact the mitochondrial metabolism of the T cells. But nonetheless, here what we found that CD38 is actually regulates breakdown NAD from the cyclic ADPR, and this cyclic ADPR is actually through the ryanodine receptor, they continuously efflux calcium from the ER. So as a result, the calcium level is all the time very high in the exhausted T cells. So we measured the calcium level and we found that yes, in the exhausted T cells, calcium level is very high as compared to the control T cells. And as soon as we knocked down CD38, it brought down the calcium level. And as I mentioned, that one of the ryanodine receptor we found, which is important for this purpose, is ryanodine receptor 2. And this ryanodine receptor 2 expression is very high in the chronically exhausted T cell of the T cells we isolated from the tumor microenvironment, both from the clinical samples and from the preclinical tumor. And we found that ryanodine receptor 2 expression is very high in the exhausted T cells. And as soon as we knock down the ryanodine receptor 2, now the T cells become good in responding to the tumor and they can control the tumor very well. So then we tried to understand what is the downstream mechanism of the calcium. And we hypothesized that the calcium, the chronic calcium level is actually important for activation of the phospho-AKT. And we measured the phospho-AKT level and we found that yes, in the chronically exhausted T cells, the AKT level is very high. And again, when we block the calcium, or we block the CD38 using a small molecule called 8-bromo-C-ADPR, the phospho-AKT level is down. And same we found using the CD38 knockdown experiment. Yes, in the chronically exhausted T cell, phospho-AKT level is very high, but when we knock down CD38, this phospho-AKT level is low. And this phospho-AKT level is actually regulates the TCF7. What we found that when we actually inhibit AKT, the cells could restore the TCF7 level. And eventually we found that yes, AKT, so the axis is actually the CD38. It regulates the NAD, it regulates the cyclic ADPR level, and when the it broke down NAD and produced a lot of cyclic ADPR, the through the ryanodine receptor 2, the calcium is released from the ER. Because of the elevated calcium level, the AKT is activated all the time in the exhausted T cells. And because of the high activation of the AKT, it actually downregulates the TCF7. And for to prove this axis, we actually block the AKT using the AKT inhibitor. And when AKT inhibitor was used, and then this combined with the anti-PD-1, the cells responded very well. So in a conclusion, what we found that the exhausted T cells, the terminally exhausted T cells, they have high expression of CD38 along with other immune checkpoints like PD-1 and TIM3. And what CD38 does, as I mentioned, that it actually elevates the cyclic ADPR level in the cells. And because of the cyclic ADPR level, the calcium level is highly elevated, which activate the AKT, downregulate the TCF7 expression. And because of the downregulated TCF1 expression, the cell failed to respond to the anti-PD-1 therapy. So as soon as we block the CD38 using the small molecule or the knockdown using the shRNA, we can actually reverse this process, improve the TCF1 expression in the T cells, and these T cells start responding to the immune checkpoint blockade therapy. So I acknowledge my lab who actually did all the work. So Annesha did most of the work, and now she is doing her postdoc in Harvard Medical School. And my lab is supported by the funding from CSR, the Wellcome Trust India Alliance, and the ACRB. And I also acknowledge the collaborators who gave thoughtful suggestions and tissue samples to carry out this important piece of work. So thank you all for your patient listening, and I'm happy to take any questions.
Right. Thank you, Silpa, for wonderful presentation. Fantastic science. May I request you to hold on to the end of the talk so we can have question and answer session at the end for both the speakers?
Sure, definitely.
All right. Thank you very much. So now we'll move on to the next speaker, Dr. Sudha Kumari. Dr. Sudha Kumari is currently Assistant Professor at Immunosurveillance, Bangalore, and at the Department of Microbiology and Cell Biology. She did her PhD from National Center for Biological Science (NCBS), especially focusing on the membrane structure and how macromolecules actually provide the structural stability to the membrane. And then followed by she went for postdoctoral fellowship in Michael Dustin's lab, New York University. And then she worked there as T-cell role of T cell structure or in autoimmunity and inflammatory reactions and immunodeficiency. And then she actually moved on to the Koch Institute of Integrative Cancer, MIT, in Dr. Darrell Irwin's lab, where she actually look at immunotherapy, how T cell contribution of T cell in and T cell interaction with other cells, and how the molecular biology of this interaction drives or contributes to the immunotherapy. And in presently at ISC Bangalore, she is looking at how these T cells or molecules on the T cell decide how to touch into the microenvironment with the other cell, and how that touch lead to the function of the cell and what dictates to it. So she has also published very well, and she got Infosys Young Investigator Award recently, and also Wellcome Trust Fellow. So without spending too much time, I'll request Dr. Sudha Kumari to share her slide, and we eagerly looking forward to listen to her how to touch makes the difference in biology, especially immunology.
Thank you so much, Professor Gadharilal. It's actually a true honor to be presenting amongst the people I have read about so much and heard from and I admire so much. So, one second, I'll just quickly find the. Oh, this is my screen sharing. Just a moment.
Yeah.
Yeah.
Probably
So
you have to select the PowerPoint.
Okay. Let me see if I can. I'll exit the presentation mode here and then try. So I've lost the habit of using Zoom, unfortunately. It's Teams at ISC. So I have it open in my computer.
If you go to share, and then probably
Share, yeah.
it will ask you which screen, and then again go to share and display.
Yeah. So once I go to entire screen, it says, it just shows basically this screen. I don't know why.
So what worked for me? So if you open the PowerPoint slide, okay.
Yeah.
And then go to the share, and then from there you just select the PowerPoint, and I think that actually worked for me.
Okay, let me
Let's
try.
learn from
Let's
what
I
works.
really It's very odd. I have the PowerPoint open here. Can I just drop it here? No. Yeah. Somehow the presentation is not showing up in the window tabs. That's the problem.
Are
The
you
full
using
screen,
Mac?
what you showed, actually, you go there and then just open your full screen on your screen itself. Only
Okay.
will able to
Just
see
me.
everything. Yeah.
Okay. Share. And then I go here. This is
Full
good?
screen. Yes. And now go to full screen on your. Yeah.
Perfect. Okay. All right.
Right. We
Thank
can
you
see
so
your
much.
slide and we can listen you. Please go ahead.
Okay. Perfect. Okay. So actually, it's my great fortune that I got to listen to Professor Shilpak, but also a little bit of misfortune because I really hope that I can still incite your interest after his great talk. So we are actually very fundamental biology lab, and we are also highly interdisciplinary. And as Professor Girdhailal mentioned, I am actually fundamentally a cell biologist, but we ask very hardcore cell biology questions in immunological context, particularly in the context of diseases, with the aim to first understand the fundamental biology, but also to hopefully find solutions to some of the diseases for immunotherapy. So what we actually study is information transfer within the first few minutes of immune cell interactions with each other, and also how these communication channels fail in diseases, and also what can be done potentially to restore these communications. So my lab, I'll just give a little bit of introduction. We actually practically started in 2022. We are still learning and setting up. But what we do, essentially do is we combine techniques from very fundamental cell biology like microscopy and reconstitution systems with tricks from immunology and with some interest in immunotherapy to arrive at understanding of something very unique to adaptive immunity, which are these very special physical contacts that they make with other cells called immunological synapses. So adaptive immunity has an important feature that it's based on recognition. It can generate memory. And it's actually quite amazing that all recognition events rely on these direct physical contacts because they are receptor ligand engagement events that dictate decision making and decisions of whether adaptive immune cells will activate or not. So we study the cell biology of the recognition and these direct physical contacts called immunological synapses. And we basically largely use cellular and subcellular systems. I will describe it a little bit better later, but we also expand these techniques to tissue level, or usually using optical techniques only, and then also at the organism level to test out the principles that we have learned at the subcellular and tissue level. So immunosurveillance, I think Professor Shilpa gave a very nice introduction to T cell surveillance and described a very nice checkpoint pathway that is implicated. But in broader sense, surveillance basically entails many processes that are very unique to immune cells. So one of them is that the T cells have to migrate long distances and infiltrate the most complex organs and physically interact with practically every cell type in the body. The cell types it cannot interact with are basically immunologically privileged because the antigen recognition won't happen there. So this is a very important, very unique thing that happens in immune cells. It's exemplified in this movie. So movie shows antigen surveillance in lymph node, which is the site of priming, and antigen presenting cells are in red and the T cells are in green. They're aggressively migrating around looking for antigens. There's no antigen here, so surveillance goes on. But if there was antigen here, the cells will stop and form a specialized conjugate interface called immunological synapse. I think all of us are familiar with immunological synapse. They are arguably the most important event, the most important structure in adaptive immune response. A lot of decision making happens at immunological synapses, whether T cell will differentiate into effector, memory, whether it'll activate or not. A lot of decision making happens in these early events. They also, in fact, target to majority of therapy, whether we realize it or not, because the engagements that are happening during synaptic formation are going to dictate and the outcome of those therapies also. But all of these steps of surveillance require this highly unique cell biology of immune cells. So just take a moment to contrast it with any other cell type in the body. So after we have fully formed, organism, cells don't really move around much, and if they do, then there's a real problem. The only cells that are exempted from this, stuck fate are immune cells because they can surveil, and also reproductive cells. So this specialized cell biology that enables surveillance is actually founded on some very special tool parts that make it possible. One is actin cytoskeleton. It's very specialized in immune cells, very different from non-immune cell types. Adhesion dynamics and their migration, infiltration, all of these cell biological properties. And there are the ones that are of great interest to me as a cell biologist. So how do we understand how these cell biological tool parts enable immunosurveillance? It's a very, very complex challenge. And it's complex challenge because of the immunosurveillance paradox. So actually, I think this paradox is very well known in cancer field. So the idea is that we actually cannot study immunes easily. In fact, there was a long debate for a long time whether immunosurveillance exists or not. And it's because it's actually experimentally invisible process. If immunosurveillance is happening in the body, you're not developing problems. So how do we know it's operating? And if the problems have already happened, what's the use of immunosurveillance? Because it cannot stop problems. So the only way to study immunosurveillance is when there is a problem. So that's what actually in the previous speaker showed, that he's studying the immunosurveillance in the context of exhaustion. So you always need a problem to actually tap into the fundamental mechanisms that enable surveillance. So you always go to diseases as a way, as a perturbation system to shed light on the fundamental mechanisms that are working in health. So it's kind of a reverse approach. And the diseases that we really like are diseases that play around with, that mess around with the T cell recognition processes, hyperreactivity, where the T cell decision making is perturbed and they react too much. And the context that we study here is rheumatoid arthritis. Hyperreactivity, when even if antigen is present, right, signals are present, but they're not reacting. And the examples are primary disease and cancer. So I will take you through one example of this approach. Through that, we will be able to appreciate how we function in the next part of my talk. And towards the end of the talk, I'll also introduce you to the projects that we are trying to set up in the lab right now. So let's start with hyperreactivity, when T cells don't function. And I've worked extensively on primary immunodeficiencies in this context. So if we have a very, very simplistic way to look at the T cell activation, T cell will engage with MHC antigen. Of course, there are co-stimulatory and inhibitory molecules here as well. This is very simplistic diagram. Leads to immunological synapse formation. There's a lot of actin remodeling there at the synapse. There's calcium flux. And somehow that leads to T cell activation. And the activated T cell can move on to perform its effector functions. So when I had started my postdoc, it was known that actin is very, very important for T cell activation, but it was not clear how. But if you think about it, I mean, affectin is basically as essential as it's a most important, one of the most important structural components of the cell. So saying affectin is important is just too obvious. It doesn't tell you how exactly it promotes T cell activation, because actin will be important even for initial adhesion, even for migration away. So what is special about actin at synapse such that it promotes calcium flux? So how do we study this relationship? How do we see how actin promotes calcium flux? So answers comes from the actin-related immunodeficiencies, where almost always there's a problem in calcium flux. So there are genetic lesions in these primary immunodeficiencies. There's a whole class of them where actin gets messed up and calcium is mixed up. So I thought this is a great perturbation context where we can actually establish the functional relationship between calcium and actin. All we need to do is choose one specific one that has very nice relationship between actin perturbation and a very well established relationship and problem in calcium, and see what exactly is happening at synapse. So of course, I was not the only brilliant one who has come up with this idea. People have already tested and sort of discarded it. So viscoelastic syndrome was my favorite one for a long time. So this arises because there's a loss of viscoelastic protein, and it's only expressed by immune cells and platelets. So when patients don't have viscotaleric protein, they develop this syndrome where T cells fail to respond. So it is a immunodeficiency, and the leading cause is mutations or loss of WASP. WASP is one of the proteins that are activated downstream of T cell receptor. When T cell receptor sees the antigen, it's activated and it causes actin polymerization. So actin is basically like a Lego. So there are a lot of Lego pieces that are free in the cytoplasm of the T cell, and you can assemble in multiple ways, and you can generate architectures and time scales of architecture, how fast, how slow. So you can really mobilize this protein to achieve any architecture and and also cellular functions related to that. So what happens when patients don't have WASP? So there is viscocelular syndrome, but there's some very interesting things that happen. One, T cells, even if you give them antigen and there's T cell receptor on the surface, they don't flux calcium. They form highly unstable synapses, and the people who have viscotaleric syndrome have very high incidence of malignancies. So I thought, great, this is a fantastic way, a natural perturbation system where we can study how calcium might participate in T cell activation, how actin dynamics might participate in calcium flux and T cell activation, how it might stabilize synapses, and how it might promote tumor immunity. So I decided to chase these phenotypes one by one to understand the basic T cell biology in this disease context. So first, I started with the calcium flux. So for that, all you need to do is first look at the synapse carefully and see what is wrong with VASP deficient T cell synapse actin such that calcium is not fluxed. So people had looked at that. So this is a classical way in which people used to visualize immunological synapse. This antigen presenting cell, peripheral antigen, and you conjugate with T cell. But turned out that whether T cell has VASP or doesn't have VASP, the amount of F actin marked by this gray staining here at synapse, which is this cell-cell junction, is similar. So people had it was a very paradoxical finding that even though VASP is missing, actin is not messed up and calcium is messed up. So it seemed like that actin was probably not participating in calcium flux here. But then these kind of assays don't really give you a good view of synapse. So I said maybe it's not the whole level of actin, like I said, rather how you're organizing it. Because Lego pieces are as good as you're organizing them. You can generate bridges, you can generate castles, you can generate trains. So organization is very, very important part of the cytoskeleton. So let's look at synapse. It's very hard to look at synapses in this kind of assays, and this is actually primary reason why not many labs across the world actually directly look at synapse. The T cells are extremely difficult optically to look at the synapses. So one way to get around that problem, so why it's so hard to look at synapses? Because the cells are very tortuous, they are round, they're highly dynamic, very small, very hard to transfect, so a lot of problems. So one way to get around that is to use the in vitro reconstitution systems. And this was pioneered by my postdoc co-mentor, Professor Michael Rustin. Basically, what you do is you make a fake antigen presenting cell. So antigen presenting cell is highly tortuous. What you do is you made a lipid bilayer and you reconstitute molecules that come from antigen presenting cell on this. And you can put T cell here, and T cell sees it as antigen presenting cell, forms synapse. Now this is a synapse that you can do a lot with. You can image it at whatever resolution you want, down to single molecule. And you can look at actin there, at great detail. And one more very important feature of these reconstitution systems is that you can precisely control the critical parameters of T cell ligand presentation here, which is the dose of ligands, affinity of ligands, because you can use altered affinity peptides, and of course you can use the diversity of ligands also you can change. So this is something that we continue to use in my lab in various different projects, and that has yielded a lot of. So you can essentially do 2D pharmacology with drugs if you wanted here, or because you can really precisely control the dose and look at the T cell output. So anyway, so in this case, we were looking at the actin dynamics. So this is what you see when you use this platform. You will get single T cell, this round thing, and we are looking at the bottom of the T cell. So T cell in synaptic state is like an egg on the hot plate. It spreads when you see from the bottom. Then this is how it looks, and we're looking at very thin, very, very synaptic area of the T cell. So actin seem to be not uniform unlike what we had seen in that previous image. It's actually very heterogeneous. This heterogeneity goes associate with the T cell receptor, so much so that they co-migrate. So this engaged T cell receptor is in red, signaling state, and then it's moving to the center of the cell. So it was very interesting to me. And then when we take T cells from VAS patient, then these heterogeneities are missing. So the synapse of a T cell from VAS patient and control and a side-by-side comparison. So now these structures we decide to call foci, and VAS deficient T cell do not have foci and they don't flux calcium. So are foci important for calcium flux, or they are just some red herrings and there's some other reason for calcium flux? So we did a lot of series of complementation assays and turned out they are important for calcium flux. What happens when T cells activate actually? So there are very few receptors that get to see the antigen. Very few antigens are generally cognate antigens are present on antigen presenting cell. So T cell has a lot of amplification mechanisms to amplify antigen signal. One of them is huge clustering of early molecules on T cell receptor cluster. And at that crowded microenvironment, what WASP does it makes these dynamic foci, which actually bring in PLC gamma 1. PLC gamma 1 is an important enzyme that catalyzes production of secondary messengers that then allow calcium flux. Turned out this was the motif that was conserved across many immunodeficiencies and not just T cells, across many adaptive cell types as well. So this was very interesting. Chasing one phenotype in an immunodeficiency led to a very fundamental understanding of how the antigen recognition leads up to calcium flux in healthy T cells. So encouraged by that, then we chased this phenotype 2, which was unstable synapses. This is actually, if you, from a cell biological point of view, extremely complex question, because you need actin everywhere. T cells have many, many modes of migration. So when they are synaptic state, they are stuck. Then they start migrating, then they infiltrate, they squeeze, they experience so many mechanical microenvironments. Many cell types that they scan have many different stiffnesses. And to accommodate all that, their cytoskeleton is highly adaptive. So how to study these adaptation? Right, so the answer will be in organization. So only once in a while you see this amazing mutation like VASP, where only one state of the T cell is affected, not other. In other words, synapse formation is not affected, migration is not affected, but only the stability is affected. And so why is stability affected? So we looked even deeper at the cytoskeleton and turned out, so this is an ultra structural view of a primary naive T cell, mouse naive T cell cytoskeleton. It's like very highly dense structure. Instead, in the VAS deficient T cells, you have a lot of holes. A cytoskeleton is basically very loose. And what ends up happening is there's a mechanical way to explain this instability problem. If you have very dense cytoskeleton, I'll not go into details of it, but there's a biophysical mechanical model that we proposed, which is that this connectivity, actually the default mode for a T cell is to walk, to always be on the go, scan, 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 scan. Most cells in our body will never get to form synapse because they will never experience the antigen. And the synapse is actually an aberration. Default is actually breaking. And so there's an active mechanism to put it to stop the T cells long enough so that they can experience the antigen and integrate the antigen signal for proper activation, and VASP enables that. When T cells don't have VASP, they break symmetry. This architecture is not generated. And even the normal T cell, they downregulate VASP to regulate the timing of the duration of the synapse. That's what we found. So two phenotypes, two very interesting results. So then we became a little more ambitious and went to the harder one. So first two phenotypes were chased in primary CD4 T cells, and phenotype 3, for obvious reasons, we switched to CD8 T cells because they can directly recognize and kill tumor cells. So high rate of malignancies probably results from many reasons, but our aim was to look at what are the mechanisms within T cells that respond to VASP and then improve the tumor immunity. So first I wanted to check whether there is any problem in VASP deficient CD8 T cells. So we took B16 mouse model. So Professor Shilpak has already introduced this model. You basically implant the tumor in flank, and if you take wild type animals or wild type as well or VASP deficient animals, so this is important to remember here that VASP is only missing from immune cells because only expressed in immune cells. there's a higher tumor growth. So there is problem here in tumor immunity, but is it from CD8 T cells? So to test that, we switched the model a little bit, and instead of VAS deficient animals, we took wild type animals and then implanted them with the B16 melanoma cells, and then do adoptive transfer with either wild type female cells or VAS deficient female cells. And VAS deficient females are significantly impaired in their ability to control tumor growth. So there is a problem in the cells. So then we did a series of experiments in signaling to map out the T cell receptor signaling to see where the problem might be. And turns out the problem is at the very early state. So contrasting with the CD4 T cells, CD8 T cell synapse is actually metastable. They form very transient synapses, and they give a lot of multiple hits to kill the tumor cells. But when they're recognizing the target, if they find antigen which is engaged with the T cell receptor shown here, they form something very interesting. They form like these structures that are similar to bacterial comet tails that if you remember from your cell biology textbooks, and that result from continuous actin polymerization. We found that was because of wasp. Wasp actually gets recruited and forms these. What do these structures do? What they do is basically they push the T cell receptor. They generate force on the bond, on the peptide MHC and T cell receptor bond. So this force is encountered by tumor cell. The tumor cell is hard; it resists the force, and this resistance actually boosts T cell receptor signaling. And more T cell receptor signaling, more VAS recruitment, more foci generation, more VAS. So the cycle is actually a positive feedback. However, if tumor cell is soft or the foci is missing, in either case we found, even if you have antigen and the right ligands on the surface, T cell fails to kill. So it's very, very interesting that the mechanical potentiation of these biochemical signals is required for T cell activation and T cell cytotoxic activity. So after that, actually, we became more and more interested in the cytotoxic T cell because of these observations to see what is the specialized cell biology they have. And we started sort of exploring even deeper. So I'm going to tell you a little bit about a surprise from my current lab that we have found an enigma and one drug. And then I'll tell you a little bit about the projects. Yeah, okay. So the surprise. So we're all familiar with the T cell image, and a lot of conferences put up, immunology conference put up the T cell images with T cells with beautiful microvilli. So after my CD8, our CD8 paper, I was very curious to know whether they're actually microvilli or there's something else. Why is this important? Actually, the T cell receptor exists on on these surface projections, and this is actually a very important cell biological feature. So what happens in the T cell recognition is speed and accuracy trade-off. Okay, so they had to be quick enough to decide whether the antigen is present and whether it's cognate or not, but they had to make accurate decision. You don't want autoreactivity, so or failure to react. So it has to be accurate at the same time. It has to be fast. So this trade-off is very hard to meet unless you have specialized mechanisms to do that. So one pathway people propose is that the presence of these surface projections, you just put all your T cell receptors for scanning there, and you screen for peptide MHC through the glycocalyx of the target cells. It also has important implication for the kind of bond you will form with peptide MHC, because there will be oscillation of the structure. It will have the mechanical pull, and right mechanical pull will give you T cell activation. So what are these structures? So actually, you can interrogate this question by going after microvilli generating proteins, or the structures that we have identified was for R2-3 dependent. So we did knockout studies in both. We made knockout animals, studied, and turns out they're neither. So they're not microvilli. That's all I can tell you right now. If you want to know more, we'll soon put a preprint on this. They're not microvilli. Very, very interesting. Then second is enigma. So this is very early T cell synapse, and this is, you're familiar with this. So actin flows towards the center. With this, it brings in the T cell receptor from the periphery, and the center is a signaling dead zone. So T cell receptor only gets to signal while it's in the movement from periphery to the center. Now jerk it, T cell line is the model of choice for doing T cell biology because they're easier, larger. And this is what people know, that most of the engaged T cell receptor goes to the center, which is degradative zone. But if you do flow cytometry, that's very contrasting. You always maintain some T cell receptor on the surface for re-engagement events. You don't want to exhaust all of your T cell receptor in the first engagement itself. So we were very curious to see where is this remaining T cell receptor going, because from the literature we know everything goes to center. Turns out everything doesn't go to center. Actually, the primary cells are very different from Jurkat, so it's also a word of caution. There are two flows: one that goes in, that takes receptor for degradation; one that goes out, that takes receptor away from degradation. So actually, primary T cells, they have a mechanism in place that rescues T cell receptor from degradation, and it's always 40%. It's almost like a gold number, no matter the antigen. So we're looking into that. But what is really paradoxical here is, how can you have two flows for the same thing at the same spot? It's like saying that a river is flowing both ways. So it's highly paradoxical. It has not been seen in any other cell type before. And third is this, that by this time we have had a chance to know many different architectures, often generated by the same molecule in the T cell synapse. And if you want to do structure function relationship between actin architectures and T cell activation molecules, we have to go beyond just looking at actin dynamics. We have to be able to perturb it, and not just perturb it, we have to perturb it when we want and where we want, right? What if I just block the whole molecule globally, I'll get rid of both architectures. I don't want that. These are Lego pieces, right? That they're two different architectures. But what I can do is, if I can inhibit in one place and not other, then I will be able to do structure function relationship. So to that and none of the existing methods do that. So for that, we have actually generated an inhibitor that does exactly that. It has spatial control, temporal control, low IC50. It has a lot of different benefits, but this is a proof of concept. So if you have a Jurkat T cell, it has synapse, a lot of polymerization and flows are happening. You shine light and immediately actin inhibition. So it's light responsive inhibitor. Without light, even though it's present, it's not going to inhibit anything. And because you can control light in space and time, you can control the inhibition in space and time, and you can ask very fundamental questions that are outstanding in the field, which is, how does early decision making when the T cell receptor is encountering the peptide MHC happens, and how does cytoskeleton assist in that? We know it assists because most immunodeficiencies also have a strong component of autoimmunity. So basically, what we continue to do is we use the diseases as a perturbation system to understand fundamental biology, with the hope that will also generate some rational design principle at some point. So going forward, the lab actually is working on three distinct lines. One is information transfer across immunological synapse, which is that given the antigen and the molecular context, what is the decision-making process that integrates the information and gives the T cell output? So it's not just purely biochemical, it's not purely biomechanical, both combine together, and they combine together to integrate the information of affinity, dose, and context, molecular context, inhibitory checkpoints are there or not. And because we can control the dose and diversity of ligand, we can ask these questions. Second is, we are studying the synapse suppression in the context of ovarian cancer, with the hope to also discover novel inhibitory mechanisms. Like Professor Shilpa talked about the checkpoint pathway that he has characterized. So are there specific checkpoint mechanisms that we still do not know about, and can we actually use them to augment existing inhibitory mechanisms? Both beyond PD-1 and beyond T cells also. So synapses are not just T cell specific, they're also, they exist in the immune cells. Can we actually augment those? And the third arm is basically manipulating T cell synapse, not through its antigen agnostic approach, not through antigen. There's so much amazing work has been done in India and beyond. I think Professor Rahul is here. I had a chance to hear his talk in ISC, very, very inspiring. So we're not doing, just from the fundamental biology point of view, what are the principles that dictate T cell activation? Can we actually play around with them using chemistry? to actually control the T cell activation events. So with that, I would like to thank this amazing team members that I have. Both PhD students as well as the master students have been through the lab over the years, and collaborators Sarit Agasti, Amit Badya at ISC, and ISC Health Center as well as Kitwai Oncology Center. Thank you so much for your time.
Thank you, Dr. Kumar. It's wonderful work and a lot of insight how they, especially T cell, decide to touch and what way they should touch to have the functional output. Now continuing to the question answer session. Now floor is open for question. And is there anyone in the panelist want to go first? All right. So if not, then I will start with the first question with Silpak. Silpak, wonderful study. Silpak, can you hear us?
Yes.
Perfect. So wonderful study where you showed that CD38 makes the decision for the T cell to work or not in terms of the exhaustion. When we look at CD38, this molecule is not specific to only T cell, right?
Yeah.
So in a tumor microenvironment, when you have a lot of cells, the signaling what you describe on the T cell, which dictates that how T cell exhaustion and the immunotherapy is going to work, especially calcium signaling. How does it operates in tumor environment, microenvironment on other cell? Is there any light on that how it operates on other cell?
Yeah, thanks Giridhari for asking this question. It's a great question. So, as you know, the CD38 is a well-known marker for the B cells. So B cells express the CD38
Right.
really high. But what people have shown in the tumor microenvironment that CD38 expression is very high on the MDSC as well. So there is a paper back in cancer research by Anil Rastogi's group from the Chicago, I guess. So they have shown that CD38 deficiency actually make the MDSC dysfunctional. And so they have shown that if you block CD38, then the MDMC will be dysfunctional. And what we have shown in this paper that CD38 is actually making the T cells terminally exhausted. And recently we have found that CD38 expression is also very high in the intratumoral Treg exclusively. So the Treg which are present in the tumor microenvironment, only they have a CD38 expression because of some metabolic differences. And those CD38 expression is not there on the Treg which are in the periphery. So that's why we believe that targeting a CD38 not only can tackle the CD8 T cell, the exhausted CD8 T cell, but at the same time can target the MDSC and mostly the intratumoral Treg, and as a result you will have a synergistic effect and improve the anti-tumor response. And if you know that CD38 is already in trial, every approved therapy for the multiple myeloma patients. So in the patients, they have not shown any toxicity related to the daratumumab treatment. So that means CD38 is not have that much of off-target effect. But obviously, that is an antibody which actually depleting antibody, so targeting this myeloma, this cancer cells. But what we are suggesting that if you can develop the small molecule that we have used in this paper, or the blocking antibody that can be combined with the different immune checkpoint like anti-PD-1 or anti-CTLA4, and that CD38 can take care of this MDSC, exhausted T cells, and that Treg in the tumor microenvironment, and overall the anti-tumor response will be boosted up.
All right. So fantastic at the way the T cells is going to respond, especially let's say if you look in the angle like adaptive cellular therapy, even for example CAR T cell or any other type of T cells, where the problem is that when they go into microenvironment, the function is not as good as the way it is expected for what you see in the hematopoietic tissues. So do you think that on those lines, CD38, when you are developing a CAR T cell or some kind of adaptive cellular therapy, might help you to modify your cells in a way that work better for the solid tumor as well?
Yes. Actually, there is a paper out recently where they have taken that strategy of targeting CD30 in the CAR T cells,
and they are making this construct where the CD38 can be targeted along with this other target. And they have reported that if you target CD38 and make the construct in such a way that actually you can improve your responsiveness of the CAR T cells for the solid tumor specifically. So our lab is also interested in that approach, and we are developing different CAR constructs in the lab, keeping CD38 in mind, one. And obviously, we have another target. We are trying to develop some bispecific CAR where we can target different molecules, and we are putting some signaling domain to modulate the CD38 expression in the CAR T cells. It's very nascent stage, so it has to go through other in vitro and in vivo testing. But the proof of concept is already there. People have shown that in the CAR T cells, if you target CD38 along with the CAR T, the like response rate will be much better in the solid cancer.
Very interesting. Is there anyone else in the panel question? Please raise your hand or just unmute yourself and then ask question. All right. So Silpak, I have one just curious to.
Sudha has raised her hand.
Oh, Sudha, please go ahead, unmute yourself and then ask question directly.
I just had a very naive question actually. So have you tried CD73 also in the context of series?
Yes, we have CD73 also. But as you know that CD73 and CD39 has to be together to make this because CD39 actually break down ATP to form the AMP, and CD73 ultimately break down AMP to produce adenosine, which is suppressive in nature. So in the T cells, we all in the exhausted T cells, so we have characterized T cells from the different tumor microenvironment, starting from ovarian cancer to breast cancer, head and neck, cervical cancer, bladder cancer. So CD39 expression on the exhausted T cell is very high. CD73 is there, but it's not to that extent of CD39 and CD38. But it is not like that. It is not at all there. It is there. But what we found predominant is CD38 and CD39 on the terminally exhausted T cells.
Okay.
And
Very
when we did the correlation study, which population is actually responding to the therapy using the different single cell data which are available, and also from our own data, single cell data, we found that there is a nice correlation of these two molecules, CD39 and 38, but we have not seen that much with the 73.
Okay. Thank you so much.
All right, very interesting. Silpak, in terms of looking at the tumor in terms of like the different grade of tumor, do you think that CD38 on T cell or in any other cell type contributes in the metastasis or progression of metastasis?
Great question again. So, although we hadn't have a chance to look into the different grade of tumor, so so in the study, what we have published, we use two different tumor types. One is TNBC and the ovarian cancer, but all the tumors what we received from the clinicians, they were in the high grade tumor. So we have not had a chance to look in or compare it to the low grade tumor. How much is the expression of CD38 on those T cells? But what we found that if the tumor is highly aggressive, if there is a report of metastasis in the patient, so we collected the primary tumor. We have not collected the metastatic tumor. We have done the primary tumor analysis, but we actually took the metadata, clinical metadata, whether the patient had a distal metastasis or not. So in those cases, we have seen that whenever there is a metastasis and the tumor is highly aggressive, this terminally exhausted T cell population is highly enriched in the tumor microenvironment. And by means of terminally exhausted T cells, I mean that those T cells express not only CD38, but also express 39, TIM3, PD1. So those population is highly enriched. But we have not had a chance to compare with the low-grade tumor. Right. How much is the expression difference?
All right. My last question to you is in terms of looking at CD38 and its role in autoimmunity. How do you think that CD38, what you discovered or what you studied in the tumor, how does it goes in terms of defining the biochemical parameters or the pathways in autoimmunity?
I have not looked into the autoimmune patients, but the student who did this CD30 work and published in PNAS, she is now in an autoimmunity lab, who have recently published a paper in Nature Communication, and they have shown this in autoimmune settings also, the cells which are exhausted and causing the pathology. So they have a CD30, they have some CD30 relation. So this paper is, I have not gone through the paper, but in a SLE patient, they have shown that a lot of pathology is driven by CD30, but the how CD30 is doing these things, so it's a open field for further investigation.
Perfect. Yeah. Thank you very much. Sudha, can you hear us? Hello? Hello?
Yes, sir, you're audible.
Oh, okay. Dr. Sudha Kumari, can you?
I can hear. Yeah. I think
Right.
I was in the mute
So,
accidentally.
no. Okay. Well, it was a fantastic talk actually to discuss about how T cell receptors on a specific location or a specific part of the T cells dictates how the function they are going to be. You mentioned to us that in autoimmune cases, when they optimally either touching, not touching, so this T cell receptor synapse, when the cells decide or the cells has to see that it is going to be tolerogenic or it's going to mount a autoimmune reaction. Can you just highlight how this contact between them dictates that are they going to be hyperactive or they are going to have tolerogenic function?
Actually, that's really good question. That sort of hits the bull's eye. So actually, there are two things that are sort of missing in the field, I would say. So one is this our extreme focus on antigen. So beyond antigen, there are many mechanisms that dictate the success of T cell activation. One is of course checkpoint, whether it's present or not. But even beyond the ligands, what is the T cell state, right? But what does it actually mean in terms of early signaling? So that question is very hard to actually ask, and you can only ask that if you can pick up the changes that are happening as the T cell is embarking on exhaustion, right? So, from the data like what Professor Silbach has shown, we have a good idea of what the T cell state is when it's intermediately exhausted or terminally exhausted. But what are the early signaling problems that actually translate into these state changes? So that's what actually we hope to pick up. And again, it has to go to a perturbation system only where we can actually try and see them. So there are two ways of do that. One is that we make a fake cancer microenvironment, bring the healthy T cell there, because this is not something we can capture, right? In solid tumor, exhaustion has already happened. What the early T cell receptor signaling events are changed, or early T cell signaling events changed beyond antigen recognition that led to this. So if we have these fake microenvironments, that's what we provide through the this antigen presentation system, we can capture some of these changes. In fact, we found that approach to be very useful when we're trying to understand synaptic suppression in some of these contexts, because in vivo you simply cannot capture them, the early events. And then the autoreactivity actually is also very interesting. So actually, if you talk to the people who do a lot of T cell systems computational analysis, they'll tell you that the system is actually designed to prevent autoimmunity more than cancer, because if you have autoimmunity, you'll die early. If you have cancer, that's a problem later. You would have, hopefully reproduced and lived your life. So it's like this whole system is designed to automate. So actually, autoreactivity is even more sort of surprise to the homeostasis. And also, how do you study the autoreactivity beyond? So one example of that is you do have self-reactive cells. So they are in the healthy individuals that do not have autoimmunity. But why do these self-reactive cells cause problem in the autoimmune individuals? So beyond the antigen recognition, what are T cell intrinsic principles that dictate this decision making? So the way we approach is we try to find again mutations that are highly prevalent in autoimmune diseases, and how these mutations actually change the early T cell receptor signaling events to basically alter the decision making. to alter the T cell output. So this is the two ways where we look at. In one case, we change the microenvironment. And actually, microenvironment also, I just want to say one more very, very interesting observation we have had is that you can present actually even the PD ligand there. Depending on what is the mechanical nature of that presentation interface, the response completely changes.
Correct.
So there's another element in the decision making that we are trying to introduce. During antigen recognition, how does the mechanical context change the...
Perfect. Silpak, you have a question. Just before you ask question, I have just continuation with the same line. Dr. Kumari, you mentioned that early detection. But if you look at in terms of immune response through developmental eyes, when the T cells are developing, they also make contact.
Yeah.
So the contact, what you see with the mature cell versus the contact, what type of contact they require during the development, is there something which is known that this dictates the molecular macromolecular connection dictates that when they decide actually now it's time to respond, and when they decide now it's time to tolerate not to respond, although contacting the same antigen?
Actually, this is such a good question. So, actually, there's a problem that people have not looked at synapses in enough context. So, I mean, synapse is not a fixed structure, and for the changes that you see in the tolerogenic versus non-tolerogenic context is going to tell you a great deal about the mechanisms that mediate tolerance. No, I don't think anybody has looked at those kind of synapses. And it's actually a technical reason, like they're just a little hard to look at, but it'll be a fantastic way to look at what are the architectures that actually generate tolerance,
Right.
and what are architectures that are more effective, like people have not really looked at those.
Perfect.
Yeah.
Silpak, yeah, you can go ahead and unmute
Yeah,
yourself. And
Can you hear me, Sudha? Yeah.
Yeah.
Yeah, it's a really, really, really nice talk. So I thoroughly enjoyed your talk. So I have two questions. So first one is a query. So where you showed this comparison between these Jarkat T cells versus the primary T cells, and you see the
Yeah.
great difference in the
Yeah.
TCR. So you used any particular antigen, or it's an anti-CD3 stimulation?
So for this particular one, we use anti-CD3. Again, that's very good question. Could be antigen specific. We do have antigen specific Jarkat system in the lab. So when we did that, it's a very sort of messed up system, but still antigen specific, NYESO specific. I have 1G4 system, and still the same response you see. So it seems same thing. So Jarkats take everything to the center, and actually then I looked at the literature for antigen-induced T cell receptor degradation. People have found differences in cell lines versus primary cells.
And the second question is that when you're talking about this early decision-making process, and so
yeah,
studies are coming up that what they're suggesting these naive T cells are not actually naive. They are already primed based on their microenvironment, or based on their what kind of signals they have encountered throughout. And as it has been reported already that cholesterol levels plays an important role in the synapse formation and TCR clustering and all these things. So have you had a chance to look into this different T cell subset, like naive T cells, primary T cells? So is there any, or do you think there could be a difference in the cholesterol metabolism, cholesterol uptake, and all these things, which is actually affecting this decision making? What would be the proper synapse, or what you are seeing in your context?
Yeah. So actually, this is again very, very good question. So I mean, the metabolic references people have shown that even the lipid profiles are different between activated and naive. Cholesterol is an important parameter in T cell decision making. That also people have shown. Only issue is that it's very hard to manipulate cholesterol in a clean way. So that has been sort of limiting and even the literature on cholesterol comes in both ways. So one way people try to show that you can make target cells stiffer, that's better for T cell recognition, you add cholesterol. But at the same time, there are studies that have shown that having excess blood cholesterol actually compromise your ability. You are actually more trigger happy as a T cell. So that literature unfortunately not very clean, but from the T cell perspective, what the few groups in China that have shown actually that T cell recognition depends on cholesterol as a cofactor. So I think low concentration, definitely it's going to augment, but at a high concentration, it starts messing up with a lot of things. And I've not actually found a good reagent to to play with cholesterol. It's very, yeah.
All right. Thank you. Thank you very much. Now I can tell you there was almost like more than 500 registrations for this webinar and 200 plus online right now watching the video. Now we have some questions in the chat box. The first question goes to Dr. Silpak Chatterjee. The question is, can activation of CD38 be helpful in autoimmune disorder?
So as I said, it's a kind of a controversial so far, because in autoimmune settings, the role of CD38 has not been properly deciphered. So people are trying to understand the role of CD38. So there is a recent paper in Nature Communication where people have shown that indeed CD38 plays an important role in the pathology, but it has not been properly described and explored in the context of autoimmunity. So, so it's hard to comment. So, how CD38 will be working in this context?
Perfect. Dr. Chiplunkar is now online. Can you just unmute yourself and ask question directly rather than I reading your question?
Yeah, I have. Can you hear me?
Yes, we can hear you.
Yeah. So great talks. Shilpak, I would like to ask you, what did you look at talks? The status of talks, and also what was the status of memory CD8 cells?
Yes, so we did look into the tox. So in the exhausted T cells, definitely tox level is very high, but we could not find any correlation or any downregulation of the tox level with the CD38 inhibition. What we found that the TCF is highly regulated by CD38, and so despite having tox, we found that despite having tox, when the T cells is restoring their TCF1 level, the cells are becoming responsive and to this checkpoint blockade therapy.
Because TCF plays a role in the differentiation of memory CD8s.
Yes.
That is the reason I asked you what is the status, what happens to memory CD8.
Memory. So we checked the memory associated gene expression, and also we actually did one experiment in the RAC deficient mice. What we did, we took the female CD30 knockout T cells or the normal T cells, normal female T cells, and then parked those T cells into the RAC deficient mice for the homeostatic proliferation. And after 21 days, we came back and rechallenged those mice with the tumor, so we found that this CD30 deficient T cells were more, the proliferate more in the RAC deficient mice when we parked them, and they expanded more when we rechallenged those mice or challenged those mice with the tumor antigen, suggesting that they have the memory potential. And when we looked into the different memory associated genes, we found a couple of the genes were upregulated, which are associated with the memory in the CD30 deficient T cells. But how CD30 is actually regulating those things, that we are recently investigating.
Okay, thank you. Now another question for Sudha Kumari.
Hi,
Professor. Sorry, Girdhari, I've been struggling between, finally I got
go
the
ahead,
Zoom link.
please. Yeah,
that is why I'm trying to call you.
No, we are in the middle of the please go ahead.
Sudha, thanks. Wonderful talk. Enjoyed it. I have a question, how does the dynamics of the actin differ when a classical APC T cell binding occurs or when a Treg interaction occurs? So, and what is the status? And did you look at the status of ICAM LFA during this?
Again, such good questions coming. So the Tregs actually I looked at when I started my postdoc, which was first cell type I studied. They make very long synapses. Their actin is extremely stable, and actually in terms of cell biology, much has been done on the helper CD4s, followed with CD8 and Tregs little, and gamma delta almost nothing. So from the Treg side of things, they seem to form very, very stable contacts, and which correlates very well with the dynamics. So that's the answer. Basically, actin dynamics is very, very stationary. In fact, based on the actin organization, you can almost. So I worked on intermediate filament cytoskeleton. Also, we have a patent on that. So that seemed to be very, very important in dictating T cell response, the Treg responses, as well as actin cytoskeleton as also. So the intermediate filament and actin cytoskeleton are closely linked there. Everything seems to be less dynamic longer synapses. So that was the case. So regarding the adhesion molecules, so we always have adhesion molecules on the on the bilayer. So there are two aspects of that. One is the amount of adhesion molecules. So there's more and more accumulating data now that suggests that actually too much adhesion is bad. People have found higher expression of ICAM1 correlating with more exhaustion, although the mechanism is not known. For my purpose, it's more the organization of the adhesion molecules rather than the amount, because on bilayer I just keep them fixed and then we just vary either presence of costimulatory ligands or presence of inhibitory ligands and just variations in antigens. So that aspect we have not studied, but actually their biology in early T cell activation is complex. So people have shown that in lymph node in germinal center reaction, actually LFA1 expression is dispensable. There are other ligands that are more important, and you are able to mount immune response against a few pathogens, and not for all pathogens though. And from the tumor context, people have shown that, the initial hypothesis was that probably the ICAM-LFA adhesions are poorer. That's why T cells are not infiltrating inside the tumor. But now there are 2-3 papers that have just recently come out that show that actually high LFA1 is bad. So I think the biology is not completely understood at this moment in the cancer context. I don't know if that answers your question.
And you mentioned about the contact, so when we in classical, when we read about the textbooks, the rolling, the tethering of T cells.
Yeah.
So how, it will be very interesting to look at those aspects when these contacts are established. So any thoughts on that?
Yeah, absolutely. So actually one of the projects I hopefully will get to share with you in the future that we're actively working on is the cytological changes that happen that tumor draining lymph node in T cells to actually attack this tumor infiltration problem only, because infiltration would require essentially two executing components. One is adhesion through LFA, ICAM1 and VLA4 and VCAM.
Okay.
And second is remodeling actin. These are two things you need. So we are trying to characterize what is wrong. Yeah. Hopefully there'll be some answers.
All right.
Thank you. Congratulations, both of you.
Yeah.
And
Thank
congrats, Girdhari, for organizing such a nice session.
Fantastic. I think, we're going to wonderful discussion and presentation, and we're running out of time now. We have some more questions on the chat box. I'll just put the question and please give very short answer. This question is from Sudha. Jyoti Shankar has put a question that we know mitochondria accumulates at immune synapse. How does TCR avidity interact with mitochondrial dynamics and mitophagy to initiate symmetrical T cell division?
Oh, highly controversial. So the mitochondrial role and metabolism has been really nicely studied, mostly by Erika Pearsall's lab. The T cell metabolic demands are of course met by the mitochondrial dynamics and localization of immunological synapse. So that happens. Now the asymmetric cell division, it's not clear whether all T cell types have that. So it has been mostly shown by, I think, Steve Reiner's lab in the context of CD8 T cells. And I'm not sure many people have consistently seen that across CD8 subsets later. So I guess that's a short answer.
All right. Thank you. Now, next question is by Nivetha to Shilpa. The question is, if we are using CD38 molecule in CAR T cell for targeting solid tumors, how the cytokine release work to kill those tumor cell, and is there any possibility of cytokine release syndrome?
So great question. So as I mentioned that there is one study where they put the CD38 in the construct and they are trying to understand that whether putting CD38 would improve the or like would means the targeting CD38. So cytokine syndrome, it's too early to comment on that part unless we have done this experiment. So we are doing CD38 construct as well in our lab. So in future we'll be able to answer this part probably in.
Perfect. One more question, the last question, from Jyoti Shankar. The question is, CD38 knockout does not alter T cell exhaustion despite being a NADS, which is the connection. What is the connection?
Yeah, so as I mentioned that CD38 doesn't alter the T cell exhaustion. So what do you mean by T cell? So what we have found that CD38 expression doesn't, or CD30 inhibition doesn't alter the expression of PD1 and couple of other checkpoint molecules, but it does affect the TCF1 level, which actually is important for different epigenetic and other signaling purposes. So that is the connection. So when you are targeting CD38, it actually, so it's a NADase, and the most important thing is that NAD regulates lot of metabolic features of the T cells. So if you have lower NAD, your mitochondrial metabolism will go bad and other metabolic things will go bad. So if you can preserve, if you can target CD38, then NAD level will be high, mitochondria will be high, and those cells will be metabolically more fit. And at the same time, the TCF1 will be high, they will be epigenetically more stable, and cells will respond to the therapy despite their having PD1 expression. So that is the whole thing, that how to improve the PD1 therapy sensitivity of these T cells. And we thought that targeting CD30 would be one way to improve their sensitivity to anti-PD1 therapy.
Yeah. Thank you very much. I think it was wonderful discussion and wonderful understanding of the subject. Both of you, what you put forward today, and we will be definitely looking forward to your future study, the new work on the same line. Thank you very much for joining us. Is Dr. Jalali is online?
Yes, I'm there.
So you want to say something about this before we close it?
Jyoti is also there. No, I just want to congratulate both the speakers and Dr. Girdhari, you too, for absolutely fascinating and very illuminating session. Even as a
Yeah.
hardcore clinician, we got some insights to understand the doubts that we had. In fact, actually opens up the plethora of questions from the clinical point and translational point to maybe this can be focused, taken into a next step in the next platform meeting as well. On behalf of everyone, great gratitude both the speakers, to the participants, amazing participation, and to all of you. Thank you.
Thank you, Dr. Jalali. And so to the audience, actually, this webinar series is the last one. We will have continue webinar series organized by the society. And the next time we look forward to you with much more strength and enthusiasm than what we have now, with some other topic related with immunotherapy with a fantastic speaker. So stay tuned. And thank you very much for all of you for joining us, and have a wonderful evening. Thank you very much. Yeah, thank you.
Thank you so much. Thank you. It was
Thank you.
really
Thank you.
educational. Thank you.
Yeah, thank you. So we close the session now.